All right. Hopefully no fire alarms today. But if there are any, I'd like to congratulate everybody on their rapid, calm evacuation of the building. And I still haven't found out if anything actually happened anywhere. But anyway, what we're going to do today is primarily talk about what's called the human microbiome. That's the collection of microbes that live in and on people. This is a type of symbiosis. We'll come back to some definitions of that a little later. And we'll continue in the next lecture also talking more about human-associated microbes, although we're going to expand beyond just those that live in and on us to those that we interact with in a variety of ways. Reminder, the midterm is Monday in the second class. If your last name is A through SAC, you will be in Freeborn Hall. If you're San through Z, you will be in Chem 179. Please get there early and bring a pen and a number 2 pencil and a photo ID. I'm going to have office hours on Friday as usual. We're going to schedule a review session. I'm not 100% sure when that's going to be yet. I'm also going to have office hours. I'm also going to have office hours today. I'm going to go just after class and stay there till about 3 or 3:30. So if you want to come then also. Anyway, since we're a tiny bit behind the original plan, we're going to do just a couple of slides to wrap up lecture 10 before we get into the human microbiome. So I was talking about extremophiles and the ways to study extremophiles and also the general ways to study microbes, and we talked about going out and doing field work on microbes, also bringing microbes back to the lab and growing them in the laboratory in something called culturing. And I went through this example of studying the biology of what are called halophilic organisms that are a form of extremophile that prefer to grow in high salt concentrations. And there's one sort of very interesting evolutionary aspect of the biology of these organisms, which is that the most extreme halophiles, the ones that can thrive and survive in the highest salt concentrations, are archaea. They have a particularly unusual adaptation of filling up the inside of their cells with salt. And those archaea that are extreme halophiles are from a single monophyletic group within the archaea. It's in the Euryarchaeota branch. There's a little bit of discussion of this lineage in chapter 26. But in theory, the extreme halophiles could have come from all over the archaea and bacteria and other groups. And it's interesting that this adaptation has basically only occurred once in evolution. There's just one monophyletic group. And then just quickly, I'm going to give you another example of extremophiles and sort of a little bit about their biology, although just a very little bit to wrap up that last lecture. So this is an overhead view of one of the more famous hot springs in Yellowstone National Park. There are microbes that live in where the water is not completely boiling along the edge, and they do all sorts of interesting functions. If you go across diverse environments and you collect microbes from places that the average temperature varies a lot, and then use those same methods that I talked about for halophiles to grow them in the laboratory, calculate their optimal growth rates using those growth curves that I showed. What people have found is that there's an incredibly wide range of optimal growth temperatures for different organisms. The optimal growth temperature usually is pretty close to the temperature experienced by the organisms in their environment, but it's not always a perfect match, and the temperature varies, of course, in most environments. So we're not looking at variation here, we're just looking at what the highest growth rate is for a particular organism. That is, where does it prefer in the laboratory to grow at what temperature? And so there are organisms that grow at very, very low temperatures. Those are called psychrophiles. There are many of them that have been isolated from deep ocean water samples where the water is very cold, or from Arctic or Antarctic environments. Organisms like us that prefer to grow in sort of middle range temperatures are called mesophiles. And organisms that grow basically, let's just say above 50 degrees Celsius, that is, they prefer to grow above 50 degrees Celsius, are called thermophiles. And as you get to really high temperatures, above 85 degrees Celsius, the organisms that prefer to grow at those temperatures are called hyperthermophiles. And there are a few bacteria that are hyperthermophiles. There are no eukaryotes that are hyperthermophiles. Hyperthermophiles, the ones that have been found, are dominated by archaea. So there's something about archaeal evolutionary history or their biology that has allowed them to, multiple species within that group, to be able to grow at incredibly high temperatures and break the monitors. If you do a comparison of all the organisms that have been characterized from diverse environments, what you basically see is that eukaryotes, there are eukaryotes that can grow at moderately high temperatures above what we survive at, but they don't push into the real thermophile, hyperthermophile range. And if you just sort of compare different organisms, archaea grow at the highest temperatures. If you find an environment that is basically above 90 degrees Celsius or above 95 degrees Celsius, the only organisms that you're going to find thriving there are archaea. And there are ecosystems where the temperature is even above 100 degrees Celsius, and that's in the bottom of the ocean where there are volcanoes and cracks in the seafloor with hot water coming up from underneath the seafloor. The water doesn't boil at 100 degrees Celsius because it's under a lot of pressure. So there are spots where the temperature of non-boiling water is something like 110 degrees Celsius, and you can find archaea in many of those environments, but you won't really find any other type of organism in those environments. Unlike with halophiles, if you map the thermophiles or extreme thermophiles even onto an evolutionary tree, what you see is that there are many examples of either individual species or individual clades within the different phyla of bacteria and within the different phyla of archaea that are thermophiles or extreme thermophiles. And what people have inferred by doing more detailed studies of these organisms and their evolution is that these represent convergent evolutionary events with separate lineages evolving the ability to grow at higher temperatures. So unlike the extreme halophiles, for extreme thermophiles, there have been multiple repeated instances where organisms have gone from being either mesophilic or slightly thermophilic to being extremely thermophilic. Many people who are either interested sort of in the general biological potential of life or are interested in isolating enzymes or processes that are tolerant to particularly extreme conditions have gone around and surveyed the biological diversity in many extreme environments. Again, as I mentioned before, in most cases, when you get to the edge of a particular environmental condition, those conditions tend to be dominated by microbes. That's not always true, but in most of the conditions it is. And so there are lots of different types of environments that people have gone to look for organisms: high temperature, low temperature, high pressure, that's like down in the bottom of the ocean, high salt, high pH, low pH, very low water levels, so organisms that are extremely desiccation resistant. There are even organisms that have been isolated that are extremely resistant to radiation. And there are many either sort of basic science uses of these organisms or applied industrial or agricultural uses. And there's just sort of a collection of different processes that have been aided by enzymes or structural components that have come from these various extremophiles. And if you're interested, there's multiple companies actually in the area that work on things like this. There's one in particular called Novozymes that's based in Davis that specializes in going around collecting or getting access to new microorganisms and isolating enzymes from them to use in various industrial or other processes. All right, so that's the end of extremophiles. Are there any questions about extremophiles before we move into the human microbiome? No? Okay. So what we're going to do now is talk about the human microbiome and also continue on this introduction to methods used to study microorganisms, and talk about what I generally refer to as CSI microbiology. You'll see what this is in a minute. And then we'll talk a little bit about different aspects of the microbes that live in and on people and how we've studied them. So if we look at this so-called CSI microbiology, the general principle is as follows. If you go to an environment, you collect a sample from that environment. Let's say it's one of these Yellowstone hot springs, but this is true for any ecosystem on the planet. And you take a sample from that and you split it in two. And you take half of the sample and you put the sample under a microscope and you count the cells that you are able to see in that sample. And you take the other half of the sample and you try to apply any and all of the methods that you have figured out for culturing organisms from that environment. So you can throw everything you have at that sample and try and grow as many things as you can from that environment. And then you count. How many were you able to grow versus how many were you able to see? And in every environment that's been looked at, you see more in the microscope than you're able to grow in the laboratory. Sometimes many orders of magnitude more. That is, you could grow 10 organisms and you might see 100,000 different things in the microscope. And what this means is basically we are missing out on understanding the biological diversity of microorganisms because, as I mentioned before, appearance, if you go out to the field and look at them in the field, it can be challenging to understand who is out there and even what they're doing from field experiments. And if you can't grow many of those organisms back in the laboratory, we've got a big gap. There are lots of things out there, and we don't know a lot about them. And in the 1980s, in particular, one researcher, Norm Pace, as well as some of his students and colleagues, figured out a solution to this problem, which was to go directly to environmental samples and isolate originally RNA and then DNA from those environmental samples, and use the same general methods that we've already talked about that Carl Woese developed and now everybody uses to characterize microbes in the laboratory, sequencing ribosomal RNA genes, for example, and apply that to environmental samples without ever having grown stuff in the laboratory. So you can go out to an environment, a surface Arctic sample or a boiling acid pool or some marsh sample, or in this case, these are these giant tube worms in the bottom of the ocean that we'll hear about more later in the class, but they function in very strange ways. They have no mouth, they have no digestive system. When they were first discovered, people cut them open and looked inside and found what appeared to be lots and lots of bacterial-like cells inside these organisms. No one could culture them, and no one could figure out what was going on in these organisms. And I'll come back to that organism again in a second. And what Norm Pace developed originally was to copy Carl Woese's method, and I'll look at the actual RNA from environments, but very rapidly people learned a new method that could be applied to studying these environmental samples to look at DNA from those environmental samples. So one of the things that Woese did was, if you remember, he extracted ribosomes from organisms and then pulled out the ribosomal RNA from those organisms. One of the reasons he did this is that organisms are filled with ribosomes. If you want to get some molecular component of a cell, a great place to get it is in a ribosome, because you can crack open cells and each cell has thousands of ribosomes in some cases. And so if you need a lot of material, which Woese needed in order to read the sequence, the string of letters inside the ribosomal RNA component of the ribosomes, he did that by getting ribosomes. But then in the 1980s, a new chemical method was invented called the polymerase chain reaction, which allows you to go to a particular organism or many organisms, and if you know a little bit of information about the gene that you're interested in, what you need is a little piece of the gene, the left side of the gene, and a little piece of the right side of the gene. You can make billions or even trillions of copies of that gene by this chemical reaction in the lab, and therefore you don't need to isolate ribosomes from organisms. All you need is their DNA. Every organism has a gene that encodes a ribosomal RNA component of the ribosome, and you can go after the DNA that codes for the ribosomal RNA rather than the ribosomal RNA itself. It turns out that this is a little easier. DNA is more stable than RNA. The method works a little better. And the way this works is great. You can go to an environment, and in a single reaction, you can make copies of all of the ribosomal RNA genes from any of the organisms in that environment using this reaction. So you don't have to know anything really about them other than that they have ribosomal RNA genes, and you can make billions and trillions of copies of them, enough to then do sequencing. And the way this works, I'm going to outline in the next couple of slides. Say you went to an environment with one species, these yellow triangles representing a particular species present in the environment. You crack them open, you isolate the DNA from those organisms. You run this PCR reaction that will make billions of copies of the ribosomal RNA genes in the genome of those organisms. So no matter what organism these are, if they're bacteria, archaea, or eukaryotes, a tiny fraction of their genome will correspond to ribosomal RNA genes, but that's the part we want because that's the part that we can compare to the ribosomal RNA genes from other organisms. You run this reaction, you make billions of copies, and then you read the sequence of those ribosomal RNA genes. Just like you read the sequence of the ribosomal RNA itself if you went after the RNA instead of the DNA. You make a data matrix where you can compare, I don't know if you can see in yellow here, the new ribosomal RNA sequence to sequences from multiple archaea, multiple eukaryotes, multiple bacteria. You build an evolutionary tree, just like we talked about in the class. And now you can say that, from this, you can say that my organism in the environment that I've never seen, I've never grown, but I know what it is because of where its ribosomal RNA sits in this tree. Let's just say that this is a cyanobacterial branch. That now tells you that that organism in that environment is a cyanobacteria. Any questions about this? General picture makes sense? If there was one organism in your environmental sample and you read many sequences, so you can do this reaction once, after your PCR you can isolate and run many different sequencing reactions of the things that come out at the end of the PCR, and all of them should basically be the same as each other, and they should show up in the same part of the tree if there was just one organism in that original sample. So this is what Norm Pace did. One of the first environments that he went to was these giant tube worms from the bottom of the ocean. Nobody could figure out what was living inside of them. Ran one of these reactions, read the sequence of their ribosomal RNA genes, built an evolutionary tree, and it turns out that these bacterial-like things that were living inside these tube worms are in fact bacteria. They're reasonably closely related to E. coli. And most importantly, they could find from this phylogenetic tree, if you compare to ribosomal RNA genes from known organisms. So when someone cultures a new species, one of the first things they tend to do is sequence the ribosomal RNA genes from those organisms. There are databases of all these ribosomal RNA genes. There are now, in fact, billions of ribosomal RNA sequences that are available. And you can figure out for a new organism what it's most closely related to that has been studied in more detail. And these bacteria that lived inside the tube worms were closely related to hydrogen sulfide utilizing chemoautotrophic bacteria that had been cultured, and lots was known about their biology. And then, even though you couldn't grow these bacteria living inside the tube worms, you could base a lot of hypotheses on organisms that they're related to that had been grown in the lab. Sort of a standard technique if you're working with plants and animals, you go to a field site, you identify a new orchid. You can base a lot of hypotheses on the fact that it's an orchid and what you know about orchid biology in order to enhance your ability to study that new species. That's what you're doing here with a phylogenetic tree of ribosomal RNA genes. Again, if there's one organism, as there was in these original tube worm samples, you should see one thing in your phylogenetic tree. If you go to an environment where there are two organisms, you isolate the DNA from everything in the environment. You run PCR, which is going to make copies of each organism's ribosomal RNA genes. And then you read the sequence of those at the end, and you should get some sequences from each organism. You now make a data matrix including both of them, build an evolutionary tree, and you can say, in this case, that that one of the organisms in your environment is an archaea, one of them is a bacteria. You can, in fact, go back into your environment and design chemical probes based upon these ribosomal RNA sequences that will stain the different organisms so you can figure out which one of them is the archaea and which one of them is the bacterium. But again, if there are two organisms in your environmental sample, you should see two things in your phylogenetic analysis. And perhaps more importantly, you should be able to estimate the relative abundance of those organisms by the number of times you see the sequence of each one. So here I've shown two red sequences, one yellow, representing the fact that the red is slightly more abundant than the yellow. You basically can do this with hundreds to thousands of sequences to get an estimate, a good estimate of the abundance of an organism. So you count 5 hits to yellow and 100 hits to red, and that gives you an estimate that red is 20-fold more abundant than the yellow lineage. Critically important for microbial studies because, again, we can't get this information from most other methods. Whereas you can go out to the field and do counts of different butterfly species or different trees or different birds, this is our way of counting, in many cases, for different microbes in the environment. If there are 4 microbes in your environmental sample, you should be able to detect 4 different ribosomal RNA sequences, placing them in 4 different parts of the tree. If there are 1,000, you should be able to tell. Also, the major limitation here is basically the cost of doing the sequencing. When I was an undergraduate, I worked in a lab studying those hydrothermal vent symbionts. I spent 1.5 years working to read the ribosomal RNA sequence from one of them, one individual organism, 1,500 or so letters of bases. It took 1.5 years and probably cost something like $20,000. And right now, in a lab at the Genome Center here, we can read about 100 million sequences for $1,000. So what this has done, the ability to do cheap DNA sequencing, which was largely made possible by things like the Human Genome Project, has allowed us to actually characterize the microbial diversity in a lot of different environments and characterize who is there and estimate what their abundance is. If we do that from multiple environments, we can then compare the lists of organisms we see, their phylogenetic positions, and their relative abundance between different environments, and now basically be able to do transects across space or do comparisons of time series for responses to disturbance, for example, for microbes, just like we've been able to do for plant and animals for many years. Just to sort of give you a non-visual text way to think about this, the general way that people characterize microbial diversity with this DNA-based method is you go to an environment, you isolate DNA directly from your environmental sample, you run this PCR reaction in a test tube in the laboratory, you take the products of that PCR and you read the string of letters in those products, the sequence. In many cases, you want to group sequences together if they're basically identical to each other, like those red sequences from the previous slide. When we group them together, we sometimes allow them to be a tiny bit different, so they might, in 1,000 bases, if they're 99.9% identical, we might call them basically the same thing. There's a technical term for this that you might see on some of my slides and in a variety of other places called OTUs, or operational taxonomic units, sort of a rough equivalent of a species. We can then measure the relative abundance of those by the counts. How many different times do we see the sequence from that particular organism? And by building an evolutionary tree, we can say what kind of organism each of those OTUs represents, as long as we have a comparison in our database of ribosomal RNA sequences. So what I'm going to do just for a couple of slides is give you some examples of what was learned, continues to be learned by this approach. So this culture-independent DNA studies completely revolutionized environmental microbiology to open up a window into understanding microbial diversity. I'm not going to go into all the details of this, just going to give you a couple of examples. And this is one in particular that was striking to a lot of people. So if you look at these evolutionary trees, this is an evolutionary tree showing some 40 phyla of bacteria that had been reported as of 2002, and some, I forget, 15 or so phyla of archaea that had been reported as of 2002. Again, the lineages we talked about in the course, proteobacteria, firmicutes, actinobacteria, they're scattered throughout here, but there are many other lineages, as I mentioned before, that we're not talking about. Yeah?
For the archaea phyla, why are there some that say bacteria?
Excellent question. So the question is, for the archaea phyla, why are there some that say bacteria at the end? Because they were named before Carl Woese discovered the archaea. So when people first isolated new organisms and grew them, they were looked at in a microscope and characterized in a variety of ways. They were prokaryotes. They didn't have a nucleus. And before Carl Woese came along, all prokaryotes were lumped together into bacteria. And only in the late 1970s, when he went and looked at ribosomal RNA sequences inside different organisms that had already been grown in the laboratory, he didn't discover those species. What he discovered was that, in fact, a subgroup of prokaryotes were in a third branch in the tree of life. The branch got named originally Archaebacteria and then renamed the archaea. But the individual lineages within those branches didn't always get renamed, unfortunately. That answer the question? And it does create massive confusion because when they rename some of these, if you do a literature search, you don't always know which name to look for. So that's one of the reasons why the halo bacteria, even though they're archaea, still have that bacteria part at the end. So if you look at these trees, each of these little wedges represents sort of the diversity within a phylum. So here's the proteobacteria. There are a lot of different types of proteobacteria. They've all been compressed into this little wedge. If the wedge is colored yellow, it means that as of 2002, at least one member of that group had been grown in the laboratory. It's a really low bar, but it means that someone somewhere had grown a representative of that phylum in the laboratory. And what's amazing from the ribosomal RNA studies that Pace and other people started to do, they started to discover entire phyla of bacteria for which no one had ever grown any member of the group in the laboratory. So each of these represents something like 1 billion years worth of evolution, and basically we knew nothing about them before ribosomal RNA surveys of the environment. And some of them are not necessarily rare. TM7 is probably in almost everybody's mouth in this room. It grows on teeth. It's a pretty important organism in terms of oral microbiology. There are many other lineages here that are reasonably abundant in different environments, but they had been completely missed because no one had been able to grow them in the laboratory. Same is true for archaea, many lineages that no one was able to grow in the laboratory, and the same is in fact true for microbial eukaryotes. We didn't show you the full diversity of microbial eukaryotes. There are also many lineages for which no one has grown representatives in the laboratory. So ribosomal RNA surveys of the environment revealed to us a much greater level of phylogenetic diversity than had been recognized previously. Drinking with the cap on. So another thing that ribosomal RNA surveys allow you to do is basically biogeography. So you're probably familiar with studies of plants and animals or other multicellular organisms where people have compared longitudinal gradients, altitudinal gradients, latitudinal gradients, comparisons across different biomes, savanna versus rainforest versus cloud forest, and so on. We would like to do the same type of thing for microbes. It's been generally hard for many of them, especially the ones that you couldn't grow in the laboratory. And these ribosomal RNA surveys opened up a whole window into not just who is out there across the whole planet, but how do they map across particular environmental gradients. And then the thing we're going to talk about, I'll explain that in a second. The thing we're going to talk about is basically treating the human body as a planet and mapping out the biogeography of the body itself and the different microbes that live in different parts of the human body. This is from a paper that was one of the first papers to delineate with these ribosomal RNA methods the diversity found in different parts of the human body and review what was known at the time. It's about 10 years old now. And so what people did is they collected samples from different parts of the human body, ran ribosomal RNA PCR, and then the pie charts show the relative abundance of different phyla. And you can see some of the phyla will be familiar. Firmicutes are the low GC gram-positives. Actinobacteria are the high GC gram-positives. Proteobacteria, there's a new phyla here that's really important in the human microbiome, and it's not one of the ones covered really in the book. They're called Bacteroidetes. And anyway, what you see is different parts of the human body have very different relative abundances of these lineages. And so they have this really nice, review paper that comes out, and they had to make the drawing anatomically correct. Now, I don't know why they had to, so I've censored it here. But if you want, you can go find it anywhere. I assume you can guess what's behind the censored marks. Anyway, so what we're going to do is go into this discussion of the human microbiome, and I just wanted to show you this. The field has been launched basically because DNA sequencing has gotten incredibly cheap. And so what I'm going to do is walk you through a couple of different ways to think about the microbes that live in and on people. The term microbiome You don't need to read all the text here. It basically is the merging together of microbiology and biome, as in like ecosystem. So this is a community of microbes that we are looking at, rather than a single microbe at a time. And what has happened in the last few years, again, largely because of the increase in DNA sequencing, is this absolute massive acceleration in a variety of studies of the microbes that live in and on people. And I'm just going to get into, because of the fire alarm, we're a tiny bit behind the other section, because I'm going to skip over a little bit of that part. So the basic general idea is every one of us is covered in a cloud of microbes in and on us. There are more microbial cells on us than there are human cells. Their total biomass is about the biomass of our brains, 2 or 3 pounds. We are an ecosystem in and of ourselves. And there are many lines of evidence that suggest or imply that this community of microbes is not just randomly traveling along with us, but they play profound roles in our biology. But the key here is that there are many suggestions that they are involved, such that we are going to say they're likely involved. But what we mean by that is that we don't really know. And I'll show you some examples of that. And we don't mean that they necessarily cause every particular human phenotype that I'm going to look at. They have some relationship to it. And I will give you some examples of strong relationships and weak relationships. So this picture basically launched the entire field. So this is an amazing study that was done by a group in Washington University in St. Louis, primarily from Jeff Gordon's lab, one researcher's lab. Peter Turnbaugh was a grad student in the lab at the time. There were many other people involved in this study. But they had this one line of mouse that it had a genetic mutation, other people had described this, in one gene that made it fat. OB, homozygous recessive for this OB gene for obesity. The wild-type mouse without that mutation were skinny. And what Jeff Gordon did was they looked at the microbial collection in the guts of these 2 types of mice. They're very different from each other. Now, that's not that surprising. The obese mouse has a lot of, inflammation going on. Who knows if it's chewing its food in the same way. It's not that surprising that its microbial community is different from the skinny mouse. What's amazing is that Jeff Gordon had in his lab a group of mice that were germ-free, that they had reared without microbes and over many generations had cleared out all of the microbes from those mice. And he took the microbes out of the obese mouse and out of the skinny mouse and transplanted them into the germ-free mice. The germ-free mice were skinny. When you take the microbes out of this mouse and put them into a genetically normal mouse without microbes, they get obese. So the microbes transmit obesity between these mice. And when this was announced, the field went insane. And everybody and their mother and brother and cousin and uncle that works on microbes started to say that they worked on microbiomes. So what I want to do is give you just a little bit of an introduction to what we now know about the microbes that live in and on humans. Again, that's mouse, not humans, but we'll talk a little bit more about humans. And the first thing I want you to sort of think about is to remember that these are communities of microbes. And if we apply concepts and tools from ecology, it can be very beneficial to understanding these communities. So the first concept that's important to think about is called biogeography. I've already talked about that here. So just like we can map the microbes across physical landscapes, we can map them across different parts of the human body. And as the technology has gotten better and better and the methods have gotten cheaper and cheaper, people have done finer and finer scale resolution biogeography of the human body. And you find that every region of the body that you can identify some major environmental difference, so elbow is really dry, other parts of the body are really wet, the skin on your elbow, the microbes there are very different than the microbes in wet areas of the body. The microbes on your forehead are different than the microbes on your chin. I mean, you are a planet in terms of the microbial diversity. And so this is like the first pass. The microbial diversity varies enormously within an individual human body, even differs on different fingers. There's some suggestion that you can use this to identify which fingers were used for touching which keys on a keyboard and even who was touching particular keys on a keyboard. So if you compare within a body, there's lots of diversity. It's also true that there's a lot of diversity between people. So this is a figure basically showing just the details don't matter that much, but the groupings here are individual samples from different people, and the closer they are to each other in this two-dimensional space, the more similar the microbes are in those two communities. And what you see is for most people, if we sampled the people in this room with a cotton swab in the mouth, and then sampled the same people in the room with a cotton swab on the skin, we would find that most of our mouth samples are more similar to each other than even my mouth sample is to my skin sample. So there's some global patterns across humans that define these individual ecosystems such that on average we're more similar between people than we are within people, different sites within people. But there is variation between people. And if you look, for example, in fecal samples or in skin samples, you find that there are different microbial communities in people with different weight classes, different body mass indices, similar to that mouse study, but it's a little bit harder to get permission to raise germ-free people. So the same type of experiment hasn't been done, but this is at least suggestive that there's enough variation between people that it might help explain some of those phenotypes. There's variation in the microbes between countries. That may be due to history, it may be due to diet, it may be due to other factors, but the people are somewhat affected by where they come from in terms of the microbial community that's found on them. Health and disease status can relate to the microbial community, so people with inflammatory bowel disease have very different microbial communities on average than people who are healthy. Again, it doesn't say that the microbes cause the inflammatory bowel disease. They could be the result of the inflammation. But certainly there is a correlation there between what microbes are found and what the health status is. And so with these sort of ecological surveys of humans across the population and within individuals, the next question you might ask is how do those differences come into being? Where do we get the microbes that are in and on us? And this is basically a question of community assembly. What are the sources of microbes and why is it that some microbes show up on the elbow and others show up in the mouth? So one important thing that has been better and better understood over the last few years is that humans are colonized. We start out sterile early in development. There is some recent evidence that you can get colonized while in the uterus, a little bit. And then people definitely get colonized when they're born by vaginal delivery. You could even view vaginal birth in part as a delivery mechanism for delivering microbes to offspring. That's what a lot of other plants and animals do. There's no reason to think that humans would be different. Once you're born, you still are a relatively open environment for microbes to colonize you. If you breastfeed, you get microbes from breast milk. You also get them from the mother's breast. And there's food in breast milk that is basically what's known as a prebiotic. It's food for microbes. There's actually a large collection of researchers here at UC Davis that work on this exact issue. There's a program called the Infant Microbiome Project, and there's also a big milk genome project in this Foods for Health initiative. And there are maybe 20 faculty who work on this issue of microbial diversity associated with breastfeeding and associated with milk. We continue to get colonized throughout our lives. Earlier in development, it's probably a little bit more important because you're a completely clean environment, but we get colonized by microbes from our buildings. We get colonized by microbes from our animals. I posted a little thing to the announcement about a blog that was written yesterday about this. This is actually in I think the front page of the Davis Enterprise yesterday also. Dogs in your houses shape the microbial community in your family. It seems in good ways most of the time. Our food definitely influences our progression of our microbial community as well. So we get colonized throughout our lives. If you're interested, my lab is starting, we figured, Many people think microbes are kind of gross. What would be better than combining a microbiome project with cute kittens? So we're starting a kitten microbiome sampling project to study the microbes on kittens. I'm happy to tell people about this in office hours or some other time. Yes, they are very cute. We compare across people. That's the biogeography. We show that they're different. There's population variability. We understand something about how people get colonized. That still doesn't tell us a lot about the health effects of the microbes. It just tells us sort of what the variation is. One really important aspect of understanding how microbes affect our health is to look at what happens when the microbial colonization or the microbial community is disturbed. This is very similar to looking at what happens when you clear-cut a forest. How does it regrow? Or when there's a forest fire. We apply the same general principles to microbial communities, and a lot has been learned about the health effects of microbes. So there are 4 major disturbance events that have been characterized in a lot of detail. Taking antibiotics, in particular when you're young, has major effects on the risk for certain health issues. So if you take antibiotics when you're very, very young, you have a higher risk of allergy, autoimmune diseases, asthma, and obesity. All 4 of those phenomenon have at least potentially a connection to microbial communities. If you're born by C-section, the microbial community, so taking antibiotics is obviously important if you're sick and you're infected by a bacterial pathogen that might kill you, but you should realize that it also severely disturbs the microbial community that you have and leads to increased risk of a variety of problems. Being born by C-section removes the vaginal colonization process that appears to be very important in humans. Kids born by C-section, at least for the first year or 2 of life, have a very different microbial community on average than kids born by vaginal delivery. C-sections do lead to a slightly elevated risk of some problems that are thought to be connected to microbes, like allergy and asthma, but it's not an incredibly high risk. It may be that you eventually get colonized by interacting with your family, even if you're born by C-section. Excessive cleanliness as a child leads to a higher risk of allergy, asthma, and autoimmune diseases. It is now thought by many people that that excessive cleanliness prevents the normal colonization of your system by microbes. And changes in diet also can be a major disturbance to your microbial community. And what all of these have in common appear to be sort of triggering miscolonization of the ecosystem, in particular when the immune system is developing. And the immune system has to learn basically to recognize good microbes from bad microbes. And it should mount a fight when you have bad microbes that are invading your system. Unfortunately, in many people, the immune system mounts a fight even when you don't have bad microbes invading the system. And that's what is basically going on with autoimmune diseases, possibly with obesity, and also possibly with allergy and asthma. And so if you disturb the microbial community, that may be why you have a higher risk of these particular problems. So we know about disturbing the microbial community in detail now because of these sort of new technologies. Of course, if you disturb something and you know that, it would be a good idea to try and restore what has been disturbed. So I'm sure many people are familiar with probiotics. Prebiotics are basically food for microbes. Probiotics are actual microbes. Taking probiotics to try and restore your microbial community is kind of like planting pineapples after you've clear-cut the tropical rainforest and think that you're going to get the rainforest back. Probiotics have been shown in many cases to be helpful, but it's an overly simplistic solution to disturbing your microbial community. Some people with severe intestinal problems that may or may not be due to microbes have gotten intestinal transplants that in some cases solve their problems, but in many cases introduce other problems. It's a very extreme response to a problem happening in your gastrointestinal system. But if you go back to this Jeff Gordon study I was mentioning at the beginning, and be prepared, I'm glad this is after lunch. One thing a lot of other organisms do is they eat poo, and they recolonize their microbial system by eating feces from a healthy individual. I'm sorry. There's something in the veterinary world called transfaunation. If you've seen the fistulated cow out by the dairy field with a big hole in its side, you can put your hand into its rumen. They can introduce hay or rumen fluid from another cow and treat many gastrointestinal problems in cattle via this method. And you can do, and people are doing, the equivalent in humans. It's called a fecal transplant. I know it sounds gross, but let's take a step back. There are many people that have what's called persistent Clostridium difficile infections, a horrible problem that can lead to weight loss and even death. The persistent infections can't be treated or prevented in many cases via antibiotic treatment. Many people who have this are just 20 years of misery. In the last few years, there has been an increase in what's called fecal transplant treatment for Clostridium difficile infection. It is exactly what it sounds like. You can take a donor material, clean it up a little bit, and transplant either via the top or via the back into the lower gastrointestinal system. Just wait. If you had this, you would appreciate it. 95% cure rate for C. diff persistent infections. It's one of the most effective medical treatments known for anything. So this is an amazing thing happening to restoring the microbial community. There's actually now attempts to create stool banks, like blood banks, to deal with this. And I'm going to leave you on one last thought, which is there are now people promoting do-it-yourself fecal transplants. There is even a YouTube video, and I will leave the video running as you walk out.